So the next speaker is Kirsty. She will be presenting on mapping African genomics.
Okay. Hi everyone, my name is Kirsty and I'm a PhD student based at UCT and also an intern at Exegetic Analytics. Today what we're going to be doing is mapping out some of the most recent genomics research in Africa, and of course we're going to use a little bit of SF. So to start off with, what do we mean when we talk about the human genome? Well, it's simply the collection of all of our genes, and our genes shape much of who we are. They influence as diverse traits as whether we can digest lactose, our ability to distinguish the color pink from the color blue, or the color green from red, and also our risk of developing heart disease or cancer. So what makes African genome data so valuable? Well, we've only just begun to understand the complexity of the human genome. We've just started identifying genes that are very strongly linked to risk. And much of this data and insight has come from European populations. The interesting thing is that African populations have an immense amount of genetic diversity, and they've undergone genetic exchange for a much longer period. So it's a big part of the puzzle that we're missing. Also, if we don't have good representation of African populations in our research, we're not going to be able to help African populations with our interventions. Okay, so now for the figure. Let's look at some recent studies exploring the human genome in African populations. Okay. So we start off with SF, of course, and dplyr and ggplot. And we read in a very commonly used data set with the world borders. We use dplyr to filter out all of the African entries, all of the features that are representative of Africa. And this is more or less what the data set looks like. So what's interesting here is that we have sub-region data. So this is what tells us North Africa or Southern Africa or East Africa and West Africa. We also have data for the country polygons and the center of the country. So I'm importing sample data. This is basically the number of participants who were sampled and their entire genomes were sequenced. So we have insight into every gene in their genome, basically. I'm just joining it to some of the coordinates that I've derived from the previous data set I showed you. Okay, this is the simple version of, I've used an automated color generator. And this is the first version of our plot. So it's just a very simple plot of all of the African data points, or all of the African countries, and shaded according to the region of Africa. So what I want to do now is I want to add the actual sampling information. I want to know which parts of which populations in Africa were sampled and what were the number of participants that were sampled. So I start with a very sort of standard, many of you will be familiar with the ggplot commands. This time I've removed the African country borders and I've added labels for the country names. I've also specified some of the map variables just for my preferences. And this is where I add my sampling locations data. As you can see, I've scaled it by magnitude or number of participants who were sampled. Okay, so this is what our plot looks like. As you can see, there are 3 data points, maybe you can see, in South Africa. The large one is, it was a sampling of the Zulu population, and this was done by the African Genome Variation Project. This is low coverage data, but they had about 100 participants. And then these 2 very small points that you might not be able to see. This was, one of them is a Sotho population, and there were only 8 participants, but it was sequenced at very high coverage, so it's much better quality data, and it gives us a lot more insight, or a lot more confidence in our insight from the genomes. We've also seen data from the 1,000 Genomes Project, which you might have heard about. The Kenya data, the Gambia data, Sierra Leone, and one of the Nigeria data points. And there are also various data points from H3 Africa. And these are all parties who've seen the value of African data and have placed investments in it. And because human genome sequencing is quite an expensive process, it's a very valuable investment. And we look forward to what we'll learn from it. Thank you very much.